All right, friends, the last chapter included in Unit 6 for AP Biology is viruses. I think they lumped it together because it kind of goes with biotechnology, but here we are. So we are going to learn what the components of a virus are, how it infects cells. There are two different cycles that it could take part in, whether it's lytic or lysogenic, and how viruses can actually introduce genetic variation, and then what we do and how it goes about doing this, I guess. And we'll also learn about differences between viruses and bacteria, which leads me to this slide right here. Here we go. So bacteria are prokaryotic cells, and viruses, on the other hand, are in that epic debate of is it alive, is it not alive, but are considered not living cells. They have genes, so they do have DNA packaged in a protein shell, but they themselves are not considered living because they cannot survive on their own. Bacteria, however, mostly are free living. Some can be parasitic, meaning they live inside of a host, but oftentimes they can live by themselves, whereas a virus strictly has to be intracellular. It cannot survive on its own. Bacteria are kind of large. A virus is about 1/1000th the size of a bacteria, so very small, which is why they infect bacteria, and the bacteria can be like little factories to make lots of viruses. Antibiotics work on bacteria. They do not work on viruses. Vaccines are used to prevent viral infections. So, as I mentioned, a virus is very small. It is even smaller than a ribosome, so very, very tiny. It has a little bit of nucleic acid, or the DNA component. It could be RNA also. So there are DNA viruses and RNA viruses. And then they also have a capsid, which is the green here, that protein coating shell. As an add-on, there are some viruses that have what's called a viral envelope that surrounds the capsid, but not all viruses do. So it's just kind of an extra piece. But all viruses have nucleic acid of some kind and a capsid. So they have a limited host range, meaning they cannot travel indefinitely outside of a host, which is what I meant when I said they can't really live by themselves. They attach themselves to the host membrane receptors. So on the capsid, or perhaps on an envelope, if they have one, they attach through the glycoproteins or something to a cell surface receptor. And then they are able to kind of inject their DNA into the host cell. They can then reproduce very quickly within the host and mature easily. Here's a picture of what a couple different examples look like. So the tobacco mosaic virus is an RNA virus. There's the adenoviruses that kind of look like this. Their influenza is one example here that does have you can see it has the envelope around it. And then I think the easiest one that we always kind of look at is a bacteriophage, which has this sheath and tail fibers and so on. There are tons and tons of viruses. Here's just an example picture of different classes of viruses based on the type of genetic material that they have, whether or not they have an envelope, and then what diseases they can cause. So just know there's a ton. And you would never have to memorize them, just more of an FYI. So what happens when a virus infects a cell? Initially, there is entry and uncoding. So the host cell might go in, the virus might go into the host cell or just injects its DNA of some kind. But essentially, you get the viral DNA. And the viral DNA is now going to replicate. It's going to copy, 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 copy. It's also going to be transcribing and making more capsid proteins. So we have viral DNA being replicated over here, and now over here we got to make those proteins to encapsulate the virus. So then they come together and they self-assemble into new viruses that will then eventually burst out of the host cell. So that's kind of the overall simplified replication cycle. They infect a host cell, they make lots of DNA, they use the ribosomes of the host to make more proteins for the capsid. They self-assemble and then burst out to go and infect other cells. There are two kinds of cycles that a virus can go through. The lytic cycle is basically the virulent or the infectious cycle. It's using the host as a mini factory to replicate, assemble, and release lots of viruses. And then the cell itself will either die by exploding, so lysing, or apoptosis, one of the two. This is active infection stage. On the flip side, there's also something called lysogenic. This is latent cycle. This means that it's not actually causing a disease at the time. It's just laying dormant. So DNA, instead of being copied and replicated and made into more viruses, is actually incorporated into the bacteria's DNA or whatever the host's DNA is. So every time the host goes to copy and replicate and become new baby cells and so on, it's copying the viral DNA too. So whether it's a prophage or a provirus, whatever it may be. And then it's possible that at some point it's laying dormant, that it could be switched on. So some kind of trigger, whether it's UV radiation or a chemical or something, is enough to click on the lytic cycle and actually make it infectious. So there's a lot of viruses that you might be infected with for a really long time without having any symptoms or any issues, and then all of a sudden it comes out of nowhere. So a bacteriophage, here's an example, is a virus that infects bacteria specifically. You can kind of see it latches on and then injects its DNA into the cell. In terms of its infectious stage, it attaches, it injects its DNA. The capsid and viral pieces are all put together, it assembles, and then eventually it bursts out of that bacteria and goes on to infect other bacteria. And that's again captured here on the left side of what happens. On the right side, you can kind of see instead of going into the lytic cycle and making more viruses, it's possible that that little DNA fragment gets incorporated into the host's DNA. That would be the lysogenic cycle. So it's laying dormant, it's not actually causing a virus at that time. Okay. Animal viruses oftentimes have that extra envelope piece. So in addition to the capsid, they have an envelope, and you can kind of see what all that looks like. But generally speaking, a lot of it is similar. You don't have to know specifics. I guess the key thing to note here is that because of that extra envelope, it makes it harder for the host organism like us to actually beat the system. Another term that might be of note is something called a retrovirus. Retroviruses use RNA. You can never really copy anything in RNA, so it always has to kind of take its RNA and turn it into DNA. That process is called reverse transcriptase, or that's the enzyme that does reverse transcription. If we remember, transcription is going from DNA to RNA. So if we need to go back the other direction, we have reverse transcription. And then the DNA is what then gets inserted into the host for the lysogenic phase, for example. Or can go into the lytic cycle and make new virus pieces. HIV is one example of a retrovirus that does this. So here you can see that when it infects a cell, we have the viral RNA And then that reverse transcription process takes place to make DNA. And then that DNA can go into the nucleus and do its thing to eventually make more virus molecules. It's kind of like just an extra step that the retrovirus needs to do. Other viruses include herpes, smallpox, and so on. Currently, we've had lots of different outbreaks. We had Zika a few years ago. Dengue fever is another. Vaccines and antivirals are used to treat viruses. Antibiotics do not work. So there's just a couple examples of real life vaccines and antivirals that we use. The last thing to talk about really is like a viroid, which is a small circular RNA molecule that can infect plants. And then it can cause errors in how the plant itself grows. So you can see here at the bottom, coconut palms in the Philippines are unable to grow correctly because of those regulatory system interruptions. Prions are also super cool because it's kind of like an infectious protein. And it was thought for a while that Alzheimer's disease is a prion disease. But essentially, a misfolded protein can actually trigger other normal healthy proteins around it to misfold. And then they start to clump together. It acts slowly, but it's kind of indestructible. Because they're clumped together, you can't even denature them with heat. So we have no cure for prion diseases. But it's pretty crazy. That's like an infectious protein. They're super fascinating. Examples include mad cow disease, Creutzfeldt-Jakob disease, Kuru. If you've ever heard of any of those, like I mentioned, it's thought that Alzheimer's disease is a prion disease because it involves amyloid plaques, so those proteins are clumped together. It's potentially thought that Parkinson's disease is the same. But all applications of viruses and infectious agents of sorts. This whole presentation, I think the key highlights are just what is the difference between lysogenic and lytic cycle and what a virus is, how it compares to a bacteria. But all of the applications are just more of a for your information versus I think that you just need to straight up know. So keep that in mind as you go through this.